The overall goal of this study is to evaluate the use of a fiberless functional near-infrared spectroscopy system to monitor frontal lobe hemodynamic and oxygenation changes induced by a cognitive task.
This method can help answer key questions about prefrontal cortex function that are most optimally studied in naturalistic settings rather than in the laboratory.
fNIRS offers the advantages of being non-invasive, portable, wearable, with minimal restrictions, make it suitable to monitor brain functional activation in freely moving subjects in naturalistic environments, such as in the centre of London.
This technique can potentially be used to detect rostral prefrontal cortex abnormalities, which reveal themselves in naturalistic tasks but not in the laboratory. It can also be applied to clinical settings like neurorehabilitation to investigate the neural effects of training procedures.
Demonstrating the procedure will be Frida Lead, a research assistant here at UCL, and Beth Swingler and Sarah Power, postgraduate students here at UCL.
Begin by setting up a camera on the experimenter's chest in order to follow the participant's movements. Then, mount a head camera onto the functional near-infrared spectroscopy, or fNIRS, shading cap, which is used to protect the device from sunlight and to track where the participant looks during the experiment. Prepare and turn on a camera for the second experimenter, who follows the first experimenter and the participant around for the entire session. Finally, clean the fNIRS headset with a sanitizing wipe. Before the experiment begins,